Hello. Just to the participants, just to let them know that we are going to wait a couple of minutes to leave time to the people to join the workshop. So in the meantime, I would like to introduce this workshop. This is dedicated to high content screening technology. The idea is to give you kind of representation of a whole pipeline from the biological aspect, and for this we will have the first talk from Nicola Gritti. He's a physicist and he spent the PhD in Amsterdam, and after a short postdoc in Jan Frisken group, he now recently joined to the EMBL group in Barcelona, to Vikas Trivedi EMBL group, and he is studying the self-organizing properties of embryonic stem cells. We are really happy to have him here, and this will be the first part of the talk about from the biological point of view and how imaging can try to address the questions of these biological aspects. Then we will move to image analysis and data mining. I will introduce the speakers when at the right time. So I think, I don't know, Nicola, if you want to start, I think I give you the stage.
Sure, thank you. So I don't remember now how I should share my screen.
Yes. You have on the lower
yes.
the lower panel, you should have a camera. Yeah.
Okay. So can you see my screen now?
It's coming, I guess.
No,
it's NG.
Okay. Okay. Yes. Now. Yes.
Okay.
Can you see it now? Yes.
Yes, perfectly.
Good. So yes, hello everybody, and thanks for the introduction, and thanks of course to the organizer for inviting us to share our idea of high content and high throughput screening. So what I would like to talk about today are two approaches to perform high content and high throughput imaging. One that is the more established and present application of HCS, and another one that is, in our opinion, what is the future and is moving towards more high throughput, high content microscopy. So first, I would like to give you a little bit of historical background. So the term high content screening historically was defined at the end of last century in 97. However, historically, it comes from several advancements that have been made in the last 50 years in, I would say, three different and complementary fields. On the one hand, there is all the engineering part, so all the definition of confocal, for instance, scanning microscopy, all the way to laser scanning microscopy, so the design of the actual microscope. On the other hand, there were advancements on the software side. So in particular, I would like to highlight the ability to perform digital imaging microscopy and the advancement in high performance computers. And finally, of course, because we are mostly interested in fluorescence microscopy, there were several advancements in the chemistry part. So the design of fluorescent physiological indicators, all the way more recently up to the discovery of fluorescent protein that allowed us to probe and visualize structure inside cells and inside biological objects. So HCS took all these concepts together and combined them in a framework with the idea of making imaging and quantification in a manner that is as automated as possible. So nowadays, HCS is a very general term that describes, I would say, a pipeline that consists of four main building blocks. On the one hand, you have the experimental design. So this is the definition of your array of conditions, which could contain single cells, but also bigger biological objects. And then there is the part on the image acquisition, which, as I already said, in the case of high content screening, should be as automated as possible and allow to perform imaging of several samples at a time. So in this domain, I would say lies what is the high throughput part of high content screening. And then the other two building blocks are, of course, once you have acquired all these images, there is all the image analysis side, which again should be as automated and as quantifiable as possible. And the quantification and data analysis part, which makes use of recent advancements in machine learning and data mining. And this, I would say, refers more to the high content part of the HCS approach. So this naturally defines a workflow in which one starts from the experimental design and ends up with analysis of your data and interpretation of your data that can make predictions and therefore can be tested with additional experiments. And so the circle is closed. So high content screening can be used in, well, two complementary approaches. One, I would say, is the fixed endpoint assays, in which it is possible to explore the space of all the possible conditions in which your biological sample is applied to in a combinatorial manner. And then for this full protocol, the samples are not imaged, and only at the last time point, samples are imaged in this high throughput and automated manner. On the other hand, one can use the same microscope to visualize cells or biological objects as they behave. And so this gives an unprecedented view on the biological process, the dynamics of the biological process, but typically at the expense that it's not possible at this point to explore all the space as much as it was possible with fixed endpoint assays. So often these two approaches are used complementarily or sequentially. So first to find the optimal conditions in which your biological sample can be grown, for instance, and then to perform a more thorough investigation of the biological process. So I would like to point out that no matter which one of the two one is interested in or is using, there is a very important challenge that one needs to address, which is that of the big data. So if you imagine having a single, in this case, for instance, 96 well plate image for several days, every 10 minutes, for instance, in two different channels, which could be, for instance, bright field and fluorescence, one ends up with already approximately 1 terabyte of data. So obviously the challenge is to manage and explore your data, to create biological knowledge from those using application softwares and informatics tools, bioinformatics software tools. So in high content screening systems, in view of the automated vision, these softwares are usually available with the systems, and so they are integrated inside the software. So the example that I would like to bring, which is the one that we use in our lab, is that of the Opera Phoenix from Perkin-Elmer. This is a very nicely looking box where basically we typically use 96 well plates that can nicely fit on the holder here. And then the moment they enter the machine, everything becomes automated and everything can be controlled with a software which is called Harmony, that is divided in 4 tabs, which represent basically the four building blocks that I talked about earlier. So in the beginning, you have the experimental setup, in which one can define different channels and different conditions of the sample. The run experiment, which is the actual acquisition, the image analysis part, and then the evaluation part. And these two are the parts that require more data analysis and image analysis. So we use this system, and I just would like to very briefly introduce you to the biological process that we are interested in. So in our lab, we study synthetic embryology. And in particular, what we do is instead of studying how different embryos, different organisms, for instance, in our case, the mouse and the zebrafish, develop in their actual maternal environment, we use embryonic stem cells, which are taken out of their maternal environment and then are cultured in a dish, so in vitro. And then we want to study what are the self-organizing properties of these embryonic stem cells. So of course, this gives a lot of benefits in terms of controlled environment, all the way to being able to perform perturbations, and of course, perform studies with high throughput. So what typically is observed when seeding these cells, roughly 300 cells in every one of these wells, is that these cells over time will aggregate. Next, they will go through a cell differentiation phase, in which, in this case, for instance, the appearance of green fluorescent signal is a proxy for a differentiation to a certain mesodermal phenotype in this case. Next, there will be a polarization event in which all these differentiated cells appear on only one side of the in vitro system. And finally, the elongation and the organization of this structure. So I would like to just give you three examples of how we use these systems to perform high content screening analysis. So, and the first one is a fixed endpoint assay, which is mainly due by Karim, who is a PhD student in the lab. So what we are interested in this case is to study the gene regulator network that is responsible for the patterning of the gastroid. And to do that, we use inhibitors, so small molecules inhibitors that are capable of perturbing this gene regulator network. And so this is a very typical fixed endpoint assay in the sense that once we seed the cells on a 96 well plate, we treat them in three different ways: either control, so the gastruloids are generated as in their standard condition, with early inhibition, which is applied between 0 and 24 hours, or with later inhibition, which is applied between 24 and 48 hours post-aggregation. And at the end time point between 72 and 96 hours post-aggregation, we simply put the plates in the HCS system, and then we image the plates with all different conditions. So once you put the plate in the system, it is, as I said, everything very automated in the sense that within the software it is possible to define the plate type. So there is a large array of plates available, but it's also possible to customize your specific plate. There are several objectives and magnifications available. It's possible to perform wide field or confocal imaging. There is a wide range of channel selections available, which makes use of the fact that the system comes with 5 laser lines and several filters that, of course, allow to image different fluorophores, which are listed in a table that can be just picked up, basically. And then, importantly, it's also possible to define the layout of the plate in case that you would like to image a fewer of all the wells that are available on the plate. So all of these normally in a customized microscope, I would say, would require a lot of manual interaction from the user with the microscope, for instance, to find the samples, to define the best settings for the imaging, and so on. And so really the idea of these systems with HCS is to make all of these as automated as possible. And if it's not possible to be automated, to make it as user-friendly as possible. And thus it allows to streamline the experiments. And importantly, it's possible to save the settings that's for your particular application and use them for future experiments. So what we observe, for instance, I will just give you an example with one of these small molecules inhibitor, which is called SHAV, which inhibits one particular part of the gene regulator network. We observe different phenotypes of these gastruloids that in a way that, for instance, in this case, in the bottom case, there is still an elongation and an organization of the structure, but it's lacking the fluorescence intensity, so the expression level of this particular gene that we're looking at. So importantly, these images can also be exported easily from the system. And so this is actually what we do. We export the images and we define an image analysis pipeline that allows us to extract the quantitative information that we are interested in. For instance, in this case, the area of the gastruloid, the eccentricity, or the anterior-posterior profile of the fluorescence intensity, which informs us on many aspects of the phenotypes that we observe. The second example is about time-lapse imaging, so more live assays. So in this case, it is also possible inside the software to define very easily a time series of events. So these, very simply, you can define, again, in which wells you want to image and how often and for how many time points. And so when we use this system to perform time-lapse microscopy of these gastruloids to understand the dynamics of the patterning of the gastruloid, we observe that there is an initial phase of growth, probably due to mostly cell division, to a phase of organization where the expression level of this particular gene comes up at high level and is polarized on one side, to a final terminal elongation of the gastruloid in the direction of the pole of expression of this gene. So again, we typically extract all this information and export all this information from all these images from the system, and then we perform image analysis on them. And for instance, we can observe that aspect ratio, so the elongation of this gastruloid, is not monotonous in the sense that it does not continuously increase, but there is a first peak which is followed by a decrease in which the gastruloid seems to again assemble in a more spherical shape, and then there is a final elongation which occurs at the time where the maximum fluorescence intensity is reached and is constant. Also, by looking at the fluorescence anterior-posterior profile, we can also look at the pattern of this expression, so that we see that in the first 80 hours of experiment, the fluorescence intensity is either non-present or homogeneously distributed throughout all the gastruloid, and then it becomes restricted to only the posterior part of the gastruloid. So this again is an example of how live imaging with HCS systems can inform you on the dynamics of the biological process of interest. So what I focus now so far is on present and very well established high content screening assays. What I would like to do in the last few minutes of the presentation is to move towards high content, high resolution imaging. So as you might have noticed in the images that I've shown you so far, those were all 2D images. It is possible, of course, with HCS to also perform 3D images. And we now know that it's essential to perform 3D images to understand the dynamics and really explore all the phenotypic variability, for instance, that is present in, for instance, in our case, in the patterning of these gastruloids. But so in the case of high content screening, because of the wide field and the confocal nature of the imaging, it is difficult in these cases to avoid phototoxicity, for instance, especially when you want to do really high timely result imaging over long-term periods. So, of course, besides HCS, there is a full optical microscopy field, advanced optical microscopy field, which makes use of additional imaging modalities. And I would like just to highlight light sheet microscopy, which is this technique which you might be familiar with, that allows you to decouple the illumination part of the imaging from the detection part, and this gives you some very nice features of your imaging device. For instance, it is now possible, because of the wide field nature of light sheet, to still perform very fast acquisition, so to really look at the dynamics of the biological process. Now, because you are illuminating basically only the focal plane that is being imaged by the detection objectives, these come with the benefit of low phototoxicity, so the biological process of interest is as unperturbed as possible. It naturally allows to move the sample in the orthogonal direction to the light sheet, so to perform 3D high-resolution imaging, and importantly, can perform microscopy across scales. So if we look at, for instance, an example of this is again a gastruloid in which there is the fluorescence intensity for genes that are expressed in 2 different regions of the gastruloid. And now using light sheet microscopy, it is possible to move inside the gastruloid and visualize and probe the gastruloid in every single cell that is inside them. So there is one thing about light sheet microscopy, which is, of course, a major characteristic of this microscopy type, is the way that the sample is maintained inside the microscope. Because of the peculiar way in which the objectives are accommodated in the microscope, the sample has to be held from the top, typically. So, and this can be achieved by either embedding your sample inside an agarose tube or holding it with a hook or inside a glass capillary, or even have it on a microscope slide that can be attached to the sample holder via a pin. So all this configuration make it a little bit more difficult for light sheet to be able to go towards the high throughput direction, because of course you cannot accommodate as many samples as you would be able to as in a 96 well plate inside an agarose tube. So I would say in comparison with more established HCS commercial system, light sheets is a little bit lagging behind in terms of throughput and also in terms of automation, both of the image acquisition and of the content, image content. However, I just would like to say that recently there have been some attempts in mitigating these two challenges. In one sense, the throughput can be improved by using objectives that are accommodated in different ways. In this particular case, the objectives are in a T configuration, so the detection happens from the bottom. And this allows you to seed your samples inside a holder, and so you can seed many of them. And so by linearly scanning this holder in the microscope, you can now image several samples at the same time. On the other hand, for image analysis and content automation, now there are a lot of open source softwares that allow you to automatically, also in an automated manner, to extract information. One of them is Cell Profiler, which is, I believe, what Hugo will talk about in the next presentation. Of course, there are other softwares such as Fiji or more custom softwares that allows you to define a custom built image analysis pipeline, such as Python. So in conclusion, this brings us to an idea that light sheet systems are becoming more throughput. Probably they will never achieve the same level of throughput as commercial system, because it will be very difficult to really accommodate as many samples inside a light sheet system. And for the content automation part, light sheet systems are quickly approaching the point where it is possible with this kind of open source softwares to perform your own image analysis pipeline in an automated fashion. So I just would like to thank everybody in our lab. Of course, all the people that have helped me put together this presentation by allowing me to show you some of the images that I've shown you, and the Microscopic Imaging Facility led by Jim, who is the responsible facility for the high content screening system from PerkinElmer that we have in the lab. And I'm happy to take any question if you have any.
Thanks a lot, Nicola. In fact, there is one question about if it would be possible to do cytometry with this kind of samples in the way that you can run this kind of screening with these automated flow cytometers.
Yes, so there are commercially available software systems definitely to perform cytometry. I'm not sure if the question regards more the commercial system or the light sheet system, but in the case of the light sheet, of course, it's a little bit more complicated because of the configuration of the objective. So typically, it would be possible in case there is a microfluidic device integrated or coming on top of the microscope, but this, of course, goes beyond. It goes more towards the custom microscope build.
Okay. Thanks, Nicola. For time reasons, I think it's better if we move, and then at the end of the workshop, we can have some more time for questions. So, as I said at the beginning, the idea is to give you a kind of general idea of the full pipeline. So now, as Nicola has presented we have acquisition and biological processes. Now we will move to the second or third leg of this chair, which is this image analysis approach. For this, Hugo Botelho will explain these image analysis possibilities. Hugo has a PhD in biophysics. He works in the Universidad Nova de Lisboa. And then he is manager of the High-Throughput screening facility at the Biosystems and Integrative Science Institute from the University of Lisboa. And he is really a really nice expert and a great expert in image analysis. So please, Hugo, could you start? Thanks a lot.
Thank you, Diego, for the introduction. Hi, everyone. So today, my goal will be to move on from where Nicola left us and address how one could do high content image analysis using some of the data sets that Nicola has presented us right now. So I will try to share my screen. So can you see my screen?
Yes.
Okay. Thank you very much. So if you want to follow along this workshop, either right now or at a later time, I've left some notes about what I'm going to show you, as well as the imaging, the data sets, and the self-profiler files in GitHub. So if you go into the description for this workshop on the SPALM website, you have the link for this GitHub repository. So the tool of choice for my presentation will be self-profiler, and for those of you who don't know, self-profiler is a free and open source software tool aimed at empowering biologists, which may or may not have formal training in image analysis and computer vision, to be able to extract quantitative features of biological images, with the particular twist that this can be done in large data sets containing hundreds or thousands of images, and this is done on an automatic fashion. So this is, of course, typically what you would do on a high content screening campaign, and on such scenarios, you may also want to extract this kind of image-based features on an per object basis, or characterize each cell, for instance, in your data set. This is done in self-profiler with a so-called pipeline, and the pipeline is a combination of basic image analysis steps, which you can configure in a way to produce the quantification you need from your data set. Self-profiler is a tool which comes from the lab of Ann Carpenter at the Broad Institute. It's based on Python and then run on Windows and Mac OS. As you will see, in the spirit of high content analysis, self-profiler implements the principle of measure everything, ask questions later. Of course, this is relevant because high content imaging data sets can be computationally intensive to analyze, so you are much better off in most cases to extract numerical features and then parse this numerical data afterwards without coming back to the image data set each time you need to interrogate your data set. One of the motivations for this workshop is that a new version of self-profiler has recently been released. This is self-profiler 4, and the current version as we speak is self-profiler 4.0.6. And most of the improvements in this version are the migration of self-profiler from Python 2 to Python 3. So most of the changes are occurring in the backend and are not so visible to the user. What it is visible is that the developers have improved the compatibility with pipelines with previous self-profiler version. So if you were using previous versions of Superfiler, most likely you will be able to import your pipeline and perform analysis on the newer version of Superfiler. Do bear in mind that some of the improvements in Superfiler may change some of the measurements you extract. So some of the libraries Superfiler is now using may slightly change some of the features you compute from images. So if you are matching results obtained with previous versions of Superfiler and this current version 4, please bear this in mind. There were also a few performance and user interface improvements, some bug fixes as well, and some of the things we will be noticing quite a bit today are the improved 3D viewer and the improved image visualization features, which are much welcome for someone who uses Superfiler on a pretty regular basis. This is the background on Superfiler, and let's move on to actually performing an image analysis pipeline with Superfiler. So the designing an image analysis pipeline. If you want to follow this along, just come to this GitHub repository. Under this green code button, just to download the contents of this repository. I have done this already, and here you will find a data set with 2D images and a set with 3D images. And inside of each data set, you'll find the raw images which we will be analyzing right now. The Superfiler pipeline files, and also the expected outputs from the analysis of this. So if you download Superfiler on its website, you can run it, and you end up with a window which looks like this once you open it. So the idea is that on the left part of the window, we will be introducing the analysis modules that we will need to analyze the data set, and on the right we will be able to configure each option. So let's maybe start with the 3D data set, since this is maybe something one that does not see often on Superfiler, the analysis of 3D data sets. And for today, Nicole has kindly shared some images from these gastruloids imaged with light sheet fluorescence microscopy. And the images we are going to be analyzing today have two fluorescence channels. So one is for the nucleus, and the one is for a GFP tag protein. Since Superfiler may not be the best tool to analyze this whole gastruloid at once, I actually took this 3D volume and selected 4 substacks. So I extracted these 4 subvolumes from this gastruloid, and the idea is that they may somehow simulate a data set with different conditions. And of relevance for us today is that these four volumes may have different expression levels of GFP. So 1, 2, and 3 may have a lower GFP expression, and the 4th may have a larger GFP signal. So to analyze this data set in Superfiler, you may come to the files you can download from GitHub and open these on Superfiler. If you do this, you'll find a pipeline which has already been configured to analyze this data set, and on each of the modules, you will find a short description which indicates what this module is implementing for this particular analysis. but our goal today is to create a pipeline from scratch. So let's clean everything and start with the clean subprofiler session. So the pipeline will be written from top to bottom. So let's go for it. So in images, I will simply drag the raw images for today's analysis. And something to bear in mind is that for a 3D analysis, the subprofiler only supports TIFF files which have one single channel. So in this case, you will see that there are the 4 substacks, channel 1 and channel 2. So these are the single channel TIFF files. We can then move forward. So in metadata, we can extract the experimental metadata from the file headers, from the file name, or from some external data source. This may be useful if you want to annotate data coming from subprofiler with information such as which plate this image comes from, which experimental treatment has been performed on these cells, and so on and so forth. So today we are not going to be extracting any metadata, and we can move forward. Next, names and types is used for teaching subprofiler how to deal with the images that we have provided. So something of relevance is that we have 3D images, and these 3D images have 2 channels with different meanings. So this is what we need to specify on this module. So we will not be addressing all images as if they were the same. Actually, each file may have different meanings depending on whether they are coming from the nuclear stain or from the GFP signal. So let's tell subprofiler that all files which end with C01.tiff are the DNA images, and we also have another kind of image which will end with C02.tiff, and these are GFP. And also of relevance, these should be processed as 3D. In case of 3D images, we need to specify relative voxel sizes in X, Y, and Z. For this particular data set, it's 0.76, 0.76, and 2. If we click update, we can rest assured that subprofiler correctly interpreted these images, where it found 4 image sets, each of them composed of 2 imaging channels. Then we may want to group images. This is typically done for time-lapse images, where groups of images belong to the same time-lapse, but today we're not going to be using groups at all. Okay, so let's start adding custom modules for this analysis. The first thing we may want to do actually is take a look at the images. So let's maybe use Fiji and open one of these images. So this is a nuclear stain, and to get information from individual cells, maybe the first step we want to add is a threshold. So let's do this. To add a module, you can either right-click and check what is available in terms of image processing, object processing, and all sorts of categories. And you may also you can also click this plus button, which lists the same information with the exception that you can actually write here and it's the available module. So let's add the threshold module. We want to threshold our DNA image, and we want to call it the outcome from this result as DNA threshold. And let's maybe use the Otsu algorithm with the default setting. At this point, we don't know exactly what will happen to this image, but we can go into test mode. And in the test mode, we can interactively see what Superfile is doing to the images. So if I click this play button here, it shows me the original image and the result from the threshold. And you can scroll this and across the side direction as this is very important. So the result is not great as you can see. Probably this has to do with this, some light scattering that we see in the raw image. So we should maybe improve or enhance this image before the threshold. So let me add. So we want to do image processing, and one of the modules is called enhance or suppress features. Let's add this. Let's add it before the threshold. Let's enhance the DNA image. Let's call this enhanced image DNA enhanced. Let's maybe enhance circles. You have to provide the feature size, and for this you can maybe, if you open an image, you have this measure length tool, and see that each nucleus is around 10 pixels in diameter. In this case, we don't have the image is not being enhanced in a meaningful manner at all. So I found that if we choose speckles and provide a slightly larger size, this should provide a good enhancement of the image. There you go. So we have an image without this light scattering. We can adjust contrast. This is an improvement in Superprofiler. These controls for interactive contrast adjustment. You can see that the image is somehow enhanced, and we can now maybe do thresholding on this image. So threshold, this time around, we don't want to threshold the raw DNA image. We would much rather want to threshold the DNA enhanced with the same parameters, maybe. It's not great. So I found that for the thresholding, the minimum cross entropy performs slightly better. Okay, at this point we only have a binary image. To be able to identify objects, we need to do some watersheding on this image to pinpoint individual nuclei. So let's watershed. The principle is the same. We want to watershed the image we just thresholded, and the output will not be an image. It will be objects. So these objects, let's call them nuclei. We need to provide some information about how this thresholding method will perform. And if you are ever in doubt, one of the great things in Superprofiler is that each field has this question mark next to it. So you can click the question mark and see what this control is doing, what this widget is doing, and in which way it will affect your image analysis. So let's click the play button. Let's do the watershed. And it has now identified this, it has thresholded the image and identified individual nuclei in this volume. Okay, at this point we already have objects, so we can start measuring densities. And if our goal is to measure GFP intensity at each nucleus, we can go to add measurement, and one of the modules is called measure object intensity. And we can ask it to measure GFP intensity at each of the nuclei it has just identified. So I will click on this button here, and let's actually do the measure. So it's showing us some statistics about a bunch of measurements it has just done for each object. And if we were happy at this point, we can just go to file processing and export to Spreadsheet. And what this does is that we can specify some location in our computer and ask it to create CSV files. So it's creating CSV files with the containing the information it has just gathered from each object. So this will be saved at the default output folder, which you can specify right here. The default output folder. Let me just select the desktop. And because I'm not interested in performing test mode again, I can just shut these eye icons so that Shopify will not be showing me each of these images as it goes through the entire data set in batch mode. And if I click Analyze Images, Shopify will be analyzing each image and generating this CSV file on my desktop. I'm already over my time, but if you go onto the GitHub repository, you will be able to find this pipeline containing some more advanced options. And these options can be, for instance, exporting this kind of images, or you can check that segmentation has been correctly performed. And later on, you can even use maybe some R script to analyze these CSV files and plot the mean GFP intensity for each of the 4 substacks, and find out that in fact substack 1, 2, and 3 have a lower GFP signal. Stack 4 has a higher GFP signal, as one can see right here. So the highest GFP signal should be found on substack number 4. So if you want to check more about these pipelines, just contact me later or check the GitHub repository. This is just to give you an example of how one could analyze a 3D data set with the Supervised.
Thanks a lot, Hugo. Really nice talk. We are running a little bit out of time, so I think it's better if we pass to the next speaker. As I said, this chair has 4 legs. We already saw image analysis and biological point of view, but there is an important step that is the data mining process. For this, and to talk about this, we have Gadea Mata. She's a mathematician and computer scientist. She has a PhD in computer science. She formerly worked at the CNIO as image analyst, and now she joins to the University of La Rioja as a lecturer. And screen is yours, Gadea.
Okay, thank you, Diego. Hi all, I'm going to share my screen. Could you see? Okay, so I'm going to explain numbers or interpretation on data analysis. That is a main part of the process of the experiment. So all of us know that an image is a matrix of numbers, and with these numbers we can, it's possible to do a lot of operation. We use some of these operations to develop algorithms to analyze the image to obtain some images, features, or descriptors, no? As Hugo showed us now. So these features are numbers too, and so it's possible to do many operations again and create new algorithms to interpret the data. In this session, we will see an example of analysis of data using machine learning techniques, and to this aim, we are going to use an Orange, that is an open source software. So we can see why I want to explain why machine learning. So we can see this graph, and where each point is a data sampler or instance. And we can imagine that each dot is an image or a cell or gastroloid, anything. So and each axis is a feature. So in this case, we have three-dimensional data. And we can guess that there are 4 groups, but they are not clear. So in fact, in this other graph, we can see that there are 4 groups, different groups, but it's not easy to split or to divide or to classify these groups in each graph. So we can use machine learning techniques to obtain these groups and to obtain any relationship that they are in among the data. Because if we think in a 2-dimensionals, maybe this task is easy using a threshold techniques, but if we think in 3-dimensionals or more, this task is more difficult. Okay, so machine learning tries to identify complex patterns in millions of data. The main goal is to develop techniques for a compute can learn, and this is the main workflow that we want to use if we want to obtain the best results. So first is we have to identify the problem. Second step is to obtain the data, to obtain the images and the analyze image. And the third step is to choose the model, which model is the best for us, for our experiment. And the last step is to evaluate and interpret the results. Okay, to explain how to use machine learning, we have to choose data set that were provided kindly by Nicola. The first one is a plate where in each well there is a gastroloid. Each gastroloid or well in the data set is an instance, and each instance has several features or descriptors. These features have been obtained from the image analysis, as Hugo has just shown us, and we know that there are 4 different conditions in this plate. The second data set is a video or time-lapse divided in 3 parts, and each part has about 60 frames, more or less. Then the possible use of machine learning is to classify the samples of this plate according to the morphology. We know a priori which are the experimental conditions, but we will not only use this information to check if our classification has some relation or not with the experimental conditions. And once we have our sample classified into 4 classes, we use the images of the video to classify each frame in the same group that the first sample. Then we have the images and we have the features, which these features describe our data. And the third point in the pipeline is to choose the algorithm to make a model to obtain a good results or our solution. So there are 2 main kinds of algorithms, unsupervised learning and supervised learning. And the first one, we don't need training. And the second one, supervised learning, they need training. And for the unsupervised learning, the main difference is that our data don't need label. And this part, if you use a supervised method, we need that our data set, they have a label. Okay? So I'm going to show how to use an example of both methods. And for the first one, the goal is to search hidden structures, patterns, or features in the data. And as I said, we don't need training when use this kind of methods. And however, supervised learning needs a labeled data set, and the model is trained with this data and for learning, for classifying. So the last step is to obtain the result and its evaluation. And to obtain a good evaluation of the process, it's necessary to have another data set for testing. Okay? In our cases, we are going to use the first data set for training the model. and the second one for testing, the time-lapse for testing. So this is the software that I want to show you. When we open Orange, we have an empty panel here, okay? And if here there is several panels with different functions, and it's very used to use. It's very easy to use because we have, for example, you want to load a file, I choose the widget, drag and drop in this part of the panel, I double click, and I have different settings of configuration that I can do it. For example, I choose a file, and if I want to see what is there in the file, I connect both widgets, and double click, and other window open with the information about this widget. In this case, the information about our file. Okay, so to go fast. Okay, this is the first part of the workflow. I want to mark that this software is very useful to visualize the data. So this panel, the visualize panel, there is different graphs and different ways to see your data. For example, if we have in this file, there is the data of the plate, the gastroloid plate. So here I can see using a box plot how is the distribution of this data. I can see all data together or separate by condition. Okay, I use here, there is the features, and in this case is the area, but I can show the equivalent diameter or the orientation, everyone thing. Okay. Other way to see the data is using a scatter plot, and this way is, I think that is very useful because you see the plate. Okay, you can see the plate if you put in the column and the row in the axis. And later, if you use the color with any features, you can see with a heat map what's meaning this feature in this place. For example, the wells in this side of the plate are smaller than this one, and only with you, I think that is very useful. Later, we can see that the data, and you can see that the features are not in the same scale. This scale, this value is bigger than this one. So to work with machine learning techniques is better than the normalized of the data. So this widget, for example, has a different techniques for normalize the data. In our case, we are standardized the data. And later, if we want to know which features are more relevant for our problem, we can use this other widget, rank, and the features are sorted for relevance. So in this case, we are only use these 5 features to simplify the problem. And once time that you normalize the data, you can see your data using other graphs, in this case a heat map. It's other way to show your data. Okay, this is a visualization data. According to the unsupervised learning, I add this part to my workflow. Okay. I select the columns that I want to use. In this case, I say that I only use these 5 features. And I put here the features that they are information, and they are help me to show in a graph or in a table, but they are not relevant for the analysis. So here there is 2 methods to unsupervised learning. I'm going to show the k-means. This is the more popular. This method divides the data using the distance. 2 elements are the same cluster if the distance is very, very, very close. Okay? They are very, very close. So it's important to choose how many clusters we want. In this case, we want 4 clusters. And in the data table, now I have the information about our data here and the cluster. For example, this row belongs to C4, cluster 4, or cluster 1, or so on. So if I so in a scatter plot, the results I can see if the cluster has a relation with the experimental conditions. No? I use the color to show the cluster and use the shape to show the experimental conditions, and I can see that, for example, the cluster 3 was there more population about the A condition. So these tools help us to interpret the data. And the last part, supervised learning. I add this part to the workflow. I have the workflow, and I put the following. I select columns to do the supervised learning. I need to the features, same than before, but now I need a target variable because this is a supervised learning. I need to label, and I'm going to train my method using this label, the cluster. The cluster we obtain in the last step. It's not necessary if we want to, if we know what condition or what cluster we want. It's only to put that feature in this box. Okay. So now, which model is better for me or better for my data? So we use in this branch the test and score, and I put the different models that are in the model panel, and connect with the test and score. And here I can see different measures that they show me if which model is the best. In this case, we are going to see the precision recall is the best if is the higher. This measure is the higher in this case in the SVM, and the precision recall has values between 0 and 1, and 1 is the best, so it's very close to the perfect result. So in this case, we are going to choose SVM, supervector machines, and we are connect with the supervector machines to my features and my data, and I need to load the new data set. In this case, I load the data of Time-Lapse, and in this case, select the features that I need, only these 5, the rest I will remove it, and put these features that they are information for me. Okay, I connect to the prediction, so the model is training here and is testing with this new data. So in prediction, I have the same information. I have the data for my new data sample, and here in this column, I have the cluster classifying after the test with the model. Other way to see it is we use a scatter plot, and we can see that this is all the frames on the video, and this is the classes that we belong it, and this is the represent with the colors. Okay, I represent with the color the SVM clusters. So if I sorry. Yeah. Okay, so some conclusions. So using unsupervised learning, we can classify our data in 4 classes according to the morphology. We use the experimental condition we know a priori to know if there is some relationship between them or the classification. And in the supervised learning, we have seen we use the field classification as to know the classes of a new data set. Each part of the video corresponds to each one classes. So here we can see how correspond the cluster with the images of the video. Okay, so I want to finish encouraging to use this tool to visualize the data and start with your projects in machine learning or data analysis. And I want to thank Diego for all support, and thank you for all your attention.
Okay, thanks Gadea, and thanks Hugo and Nicola. So I think we have time for a couple of questions. I will ask the first one, Hugo. You know, this kind of discussion about the deep learning approach and this kind of pipelines of order, these traditional image analysis routines that are updated every day. So do you mind to discuss a little bit about this kind of comparisons, and you, Gadea, and Nicola also, about this? Which will be the future? To move all to deep learning stuff, or to keep this old-fashioned and so nice technique of image analysis routines?
Yes. I think it's almost nobody questions that AI-based approaches, such as machine and deep learning, are the future in the field of image analysis. Actually, in the dataset I showed you today, one of my greatest challenges was actually to achieve a proper nuclei segmentation. It's one of the most basic things in image analysis. And I think that the very dataset I demonstrated today would pretty much benefit from some sort of deep learning approach, either for nuclei segmentation or just for image restoration. We must not forget that there are multiple applications of deep learning. You can still use, you can maybe still use a traditional thresholding method, but with images restored with the deep learning based approaches. So the performance we can get from using deep learning in image analysis can be quite amazing, but then it may not be straightforward to most users. It's one of the greatest hurdles is how to have users or even by image analysts which are not which have not been trained ab initio in computer science to use these deep learning approaches in their day-to-day. The result can be amazing, but there is a steep learning curve which may have to be overcome depending on the application.
Nicola, do you want to add something, because you use deep learning in your analysis, right?
Yeah, so I think there is no absolute yes and no answer in when deciding between more standard image analysis approach and deep learning. So, well, one of the, I think we have seen in one of the previous talk in the main stage about this new platform to perform deep learning in a very simple with Colab. So I think now it is really moving in the direction that it will be accessible to wider range of image analysts, biologists, and experimentalists. I have used it to do image restoration, for instance, or segmentation. I think it very much depends on the application. In the case of the 3D images that we have looked at today, I think maybe a deep learning approach with image restoration would be beneficial in order to increase the signal-to-noise ratio of the cells. Because, for instance, in that case, there is a very nonlinear dependence of the scattering of the signal-to-noise ratio as you go deeper into the tissue. In this case, the gastrolite. And these depend on so many factors that it could be beneficial indeed to use a deep learning approach.
Okay. Gadea, about data mining and machine learning in data mining, do you which will be the recommendations that you would give to a new guy that enters in this field?
Well, I think that the machine learning is okay. Deep learning is a part of the machine learning, so everything solution that you can give from deep learning is inside of machine learning. So I don't know. I think that the field has to evolution, and I'm not sure about that answer about this.
Okay. Okay. We are running out of time, so Hugo posted already the GitHub information for following his image analysis pipeline. I don't know if you have any question, or if not, we can finish. I would like only to thank all of you for attending the workshop and the speakers that made a great job. And thanks a lot for joining and for being there. Thanks a lot. See you soon.
Thank you.
Bye.
Bye, thank you.
Bye.